Hey guys, welcome to my YouTube channel. In this video, I'll discuss about the topic regarding biotechnology principles and its process. Biotechnology. Biotechnology is the process. Bio means like using the living organisms or enzymes from organisms to produce something product like something which is useful for humans. By using some kind of living organisms like enzymes, uh, living organisms, it producing some kind of by use of technology, it produces some useful process to the humans. So in this process, making curd, bread, wine, which all are microbe mediated process. These are the mediated things are useful for the biotechnological process. So here, however, it is used in restricted sense, refer to those process which are useful for the it also includes a genetically modified organism. So in small, in a limited time, you due to presence of the some single enzymes, it produces the large scale product is known as the, by use of the living organism, is known as the biotechnology. So further, many other process and technologies are included under biotechnology. Example, in vitro fertilization, test tube babies, and some vaccines producing. These are all part of the biotechnology. So the EFB is nothing but European Federation of Biotechnology. It's given the definition of biotechnology, that the traditional view and the modern molecular it is nothing but the integration of natural science and organism uh, parts thereof the molecular analysis for products and services. So simply biotechnology means using bacteria or any other enzymes or fungi, using those to produce some useful product in a large scale, is known as the biotechnological process. Here the principles here the principles of the biotechnology. So biotechnology is of modern biotechnology is of the two types, which includes genetic engineering and microprocess engineering. So genetic engineering includes, which includes the genes like DNA, RNA, genetic material. By using genetic material DNA, RNA, so these genes can be taken out and introduced in the host organism, so they can be seen the changes in the phenotype of the organism. So this process is known as the genetic engineering. It is widely used in DNA or RNA, uh, like vaccines and other process. Genetic engineering is widely used. And next, coming to the bioprocess engineering. So maintenance of the sterile, microbial contamination free, which means free from bacteria or any other microorganisms. So these process enable growth of the only desirable micro eukaryotic cell in large quantity for manufacture of the biotechnological products like antibiotics, vaccines, and enzymes. So recently, the COVID vaccine, Covaxin, or regarding any Covishield, by bioprocess engineering. So they can be sterile, no other microorganisms unable to enter into that process. So they manufactured into very large quantity for manufacture of the biotechnical products like antibiotics in a large scale. So so by the advantages of the sexual reproduction over sexual reproduction. So asexual reproduction, coming to this, one parent gene, parent and daughter cells are very similar in nature. When it coming to asexual sexual reproduction, the variations can be seen. So Like asexual reproduction preserves the genetic information, while sexual reproduction permits the variation. uh, Asexual reproduction, the parent and the daughter cells are similar in nature through phenotypic in nature and genotypic in nature. While coming to sexual reproduction, variation can be seen in it. Traditional hybridization procedure used in plant and animal breeding very often lead to the incursion multiplications of the undesirable genes along with desired genes. The techniques of the genetic engineering includes like recombination DNA, so gene transfer and gene cloning. So these are the things will be used for the gene cloning, gene transfer and recombinant DNA. So to introduce some other place in a chromosome at a specific location or specific sequence, there is a place that is known as the ORI. It is nothing but the origin of the replication. To introduce into a host organism, origin of replication can be done. After that origin of replication, cloning is done and introduced into a plasmid. Then after that, the transfer of the gene is takes place. So up to here we'll discussed about the what is the meant by the biotechnology, what are the uses of biotechnology and different types of the biotechnology like genetic engineering and bioprocess engineering. How the biotechnology is used in two different types of the reproduction like asexual reproduction and sexual reproduction. So after that, different kinds of the technique genetic engineering is seen like recombinant DNA, gene transfer, and then after to introduce at specific location in a chromosome at a specific form, ORA can be seen, that is meant by the origin of replication. So in the further classes we'll discuss about the regarding from origin of replication to transfer of the gene. So if you like this video, please like, share and subscribe. Thank you.